So our last topic of the macromolecule is a protein. So up until now we have looked at three different such molecule of carbohydrates. I like carbohydrates to try to sum up what we have so far is the carbohydrate. Once again, a polymer, or if carbohydrate is a polymer, then it should be a polymer of a monosaccharide. Okay, always that such relationship exists, monomer, polymer, and through chemical bond, in particular covalent bond, more in particular that condensation reaction. In the case of carbohydrates, monosaccharide is the smallest individual unit through by putting each individual monosaccharide together to build more complex one. Then what about the function? Usually their functions are energy, immediate source of energy by breaking them down, or storage form of energy, or in some occasions structural, protective form of structural form. That's their usage in our body. In the case of lipids, lipid is also how can we describe overall lipids? In the occasion of fats and phospholipids too, to some degree, they are polymers of what? Fatty acid. Fatty acid built together to form fat. And phospholipids, if you try to remember, is a actually special kind of a fat. Usually, to become a fat, you have such a lagging. I'd rather not use this. Oh, so it's counterproductive. Sorry. Any fat, you have this basic module, okay, to which you hook 1, 2, 3. Each of this line strand represent fatty acid. Then you have one whole fat. Whether it is a fat or oil simply is determined by these guys. Whether you have this double bond, so unsaturated, then it will become oil. Or if each of these fatty acids are highly saturated, that means no double bond, then it is a solid like fat. That's it. But in the case of a phospholipid, what happens is this guy is missing. Okay. You have only two fatty acid chain, but where has this the last fatty acid has gone? Because you don't have any more room, because this plug is being used in another connection, which is phosphate. And to make matters worse, this phosphate also bring some other more hydrophilic. So that's basically the basic structural scheme of phospholipid. That's why I say phospholipids too, it is a kind of a special type of fat. Because you build the phospholipid basically using a fatty acid assembly. You don't really know all of these details, but if you are kind of confused, why do I have so many different complex schemes? And that's my effort to explain a little like a simpler terms. But anyway, more importantly, phospholipids, in the case of phospholipid. Why? Why and how phospholipid can be used as biological membrane is actually the most important information for us. And hopefully all of us understand why and what kind of feature, what kind of special feature present in the phospholipid enabled these phospholipids behave or serve as building block for such important structure, which is a cell membrane. That's all we need. And in the case of DNA and RNA, the nucleic acid. Okay. What's the concept of individual nucleotide that we need to understand? What's nucleotide? How do we make any nucleotide? Simply put, nucleotide is nothing but three components put together. One component is a sugar, pentagon-like sugar. Another component is something like we call the base. Why? Because this position, you can have a freedom of four alternative different kind of chemical structure of bases. Whether we just simply abbreviate this area as either A or G or T or C in the case of DNA. In the case of RNA, A, G, or U, or C. So probably all of us notice that the only difference here is T and U. Everybody else is the same. Whether it is a DNA or RNA, AGC, all present in DNA as well as in RNA. But T only, this special type of base T can only come for DNA, whereas this, whatever it is, the base U can come only for RNA. That's the difference. And there is a phosphate. Okay. And what else? What else? What kind of other difference between DNA and RNA did we go over? The only other difference between DNA and RNA is this position. This position, whether if you have this position, no oxygen but simply hydrogen, so oxygen is missing, okay, implying that, so that's the name, the DNA came, deoxy, oxygen is missing. Then we recognize that as a DNA making nucleotide. On the other hand, if this position, instead of a simple hydrogen, but oxygen and hydrogen, so there is an oxygen present, then we call that as a not deoxy, no, simply RNA, because it is particular name of this sugar is ribose. So ribose sugar containing nucleic acid, so RNA we call it. Actually, this RNA is more original, older than DNA in terms of this originality. Later, maybe in the next chapter, probably we have an occasion to go over this, whether which one came first, whether it's the RNA or DNA. Maybe some of you thought DNA was like a forefather and RNA is a kind of byproduct. No, it wasn't. There, in the beginning, RNA was, and DNA was actually a newcomer, and then took over original position of RNA as a genetic material. Funny thing, but not today. So that's the basic structure of a nucleotide. What's nucleotide? What's the significance of a nucleotide? Because it is an individual component. It is an individual monomer. In the case of building the special type of a polymer we call nucleic acid. Okay. So always this relationship exists. Monomer toward polymer. There is only way. If you want to build a polymer starting from small monomer, there is only one way. You establish some specific covalent bond we call a condensation bond. That's it. Okay. And more important feature about this nucleic acid is their ability of complementary base pairing. Complementary. And the basic mechanism behind the complementary base pairing is what? That's another chemical bond, which is hydrogen bond. Did I mention that hydrogen bond is very weak? Yeah. It is no match. You cannot compare the strengths of a hydrogen bond with more stable covalent bond. Even so, such a long DNA strand is maintained. is being held together using only hydrogen bond, because hydrogen bond is extremely expensive. In mass, okay, all together, hydrogen bond strength is not negligible, but really, really significant. And also, flexibility is also another the plus. Unlike covalent bond, this hydrogen bond is very flexible so that you can easily break and then reform, just like the case of water. So that's the benefit of using hydrogen bond in the DNA, double strand. And another thing is complementary things, because the difference is in the number of hydrogen bonds they can form. G and C, whatever those base, between G and C, 3 hydrogen bonds are possible. Between A and T, only 2 hydrogen bonds are possible. If that was the case, you have some intrinsic separation among these 4 different nucleotides. That's why I brought up the example of these 2 incompatible plug, electrical plug. Here we have 3 plug electrical, the code, power adapter. And somewhere else in the world, there are only 2 plug things. So if you bring these 3 plug things, it doesn't fit like relationship exists. And that makes this association, the relationship, so specific. Always between G and C and A and T. Okay, exclusive. And the significance, what? Complementary base pairing significance, it is very important. Genetic material. That's what made this DNA to serve as the genetic material. Why? Because they know then, if they know that they always work in this complementary base pairing rule, then they always know what is my partner. So they can easily pick up their respective partner in building another new synthesis of the such DNA strand from existing one. So this DNA replication can occur from already existing old DNA strand, simply split, and then new strands are synthesized alongside. How? Because they can also have a new complementary base pair relationship. That's all. Okay. So that's kind of an essence, like a keynote of all those 3 different polymers. I mean, the organic macromolecules. The last one, proteins. So we also go over the same type of this rules of recognizing, like, what is their respective monomer and blah, blah, blah. And that's the usual standard way of studying this protein in the general biology. So like it begins with proteins are polymers of amino acid, which means amino acid, individual amino acids, just like the case of individual nucleotide assembled together to build long polymer chain of DNA. Like in the case of protein, simply this individual, whatever it is, the amino acid, individual ones, got together to have this specific chemical bond. Is that going to be the same old condensation reaction? Of course, yes. This is another same old condensation reaction. So water molecule will be out, and although their respective names will be different to recognize to distinguish differences, but basically same. In principle wise, they are the same. So these amino acids build together so these peptide bonds are their special name of condensation reactions, but who cares? Yeah. That's the key. And so how many of these such amino acids are there? Whatever. This is basic chemical structure of amino acid. If you were taking the general biology, of course I would require every one of you to at least know this basic structure, but not here. So simply suffice to say that any different amino acid as a monomer of protein has a very unique, distinct organization or structural scheme. Is that all you know? Simply, that's it. How many of them? 20 different amino acids are there. That's it. So in the case of DNA or RNA, the different kind of nucleotides are only 4. But in the case of protein, you can have a more variety of different protein synthesized because what? 20 out of 20, you have a choice. Like let's say, try to compare. Let's say how many different, once again, how many different ways you can create different nucleic acid made of 100 nucleotides versus how many different ways of making a protein made of 100 different amino acids. If you calculate, like as if you were a math student back in high school, it will be like heaven and earth differences because out of 20 you are trying to pick one versus out of 4 you are trying to pick one will be huge differences. What's the big deal? So protein, that is the reason why proteins are diversity. In case proteins can be made in so many various different structural shapes. Why? Because you have such more freedom of choices, 20 different amino acids. So that's why proteins are so unique. Each individual protein can become very unique in structure because so many different amino acids can come. Actually, in the world, the number of different amino acids are not only 20. Perhaps there are maybe even more than 100 or even several hundred different amino acids you can think of. But luckily, thank God, in our body system, in living system, only 20 of such different amino acids somehow decided to join in this silly business of protein making us a little getting little confused about it. That's maybe a better. Uh, maybe that's just a blessing. Anyway, important point is diversity. This figure illustrates the place of protein synthesis. Unlike the rest of the other 3 different biological macromolecules, proteins, the location for protein synthesis is very unique in that it can occur only in one place, which is, can anyone recognize this funny structure? This is ribosome. Whenever see this something like a zome, then probably you prepare to get to see a big thing in the cell. A zome means something very big. So ribosome is something which is related with probably the RNA, I guess. Yes, that's what it is. But the only important thing is this whole structure, ribosome is the only place where protein can be built in the cell. Like carbohydrate can be synthesized anywhere actually. Whereas as long as enzymes are there. DNA or RNA can actually basically, although basically DNA and RNAs are usually synthesized inside of the nucleus in the eukaryote cells, but there is no limitation of where there is, as long as there is some specific enzymes, then DNA or RNA can be built. But protein, the lipids also. Lipid can be synthesized anywhere as long as some respective enzymes are present anywhere in the cell. But for this protein synthesis, probably you can say that for protein too, as long as the ribosomes are there, the protein can be made. But the important point is this ribosome is the very specific, unique place where protein synthesis can occur. Nowhere else. Without ribosome, there is no way you can synthesize the protein in the cell. Of course, in the laboratory, you can even try to synthesize the protein in the test tube. But in that occasion, even the ribosomes are actually really a lot of times it's needed. And what else? So this diverse shape and the wide range of functions are also associated. Shape dictate function. The life is not really sophisticated. Something like the engineered machine, it is not like such that. So the reason why a certain magic works in life is simply because it has such respective shape of that related structure. So always remember, shape dictate function, and that is the reason why different type of tissue cells have a different shape. Very good example is your nerve cell, neuron cells. Okay. The reason why neuron cells has a so very unique shape in comparison with other regular animal cell is because neuron cell has a respective, very specific own function, and to fit into such function, you have to have such a adapted shape like that. So that's where proteins are like this enzyme magicians. All our activities are actually cannot be carried out without enzymes, and that explains why to become an enzyme. Well, everybody wants to become an enzyme because everybody wants to play an important role. Okay, then how can I? If you ask yourself, how can I become an enzyme? Then perhaps the answer, 9 out of 10, the answer would be then you need to become protein first. That's what enzymes are almost exclusively. Enzymes are made of proteins. There are some few exceptions though, but I'm not going to go over that here, the exceptions. But yeah, we can safely say proteins are the one actually make enzyme. Not only the enzyme, but also other important functionally and structurally in the cell for the activity of cells, like including receptors. I have introduced the concept of receptors several occasions already. And similar to receptors, but doing a little bit different function, transporters. Okay. Can you tell the difference between transporters and receptors, function-wise? How can you? How transporters are different from receptors functionally? Receptors usually, as a result of this interaction, receptors are usually transmit some information, signals. Okay, I've got you, kind of information. Whereas transporters are the one actually allowing the passage of that respective the molecule, like glucose receptors. I mean, glucose receptors in imagination, glucose receptors would do such function of like, okay, I recognize the presence of glucose, and then I will deliver this signal information to the nucleus. Okay, we have a plenty of glucose. That's what usually the receptors do. Whereas glucose transporters actually will allow the traffic of glucose through membrane. That's the difference between transporter and receptors. And everybody now in these days of a pandemics, everybody appreciate the importance of the antibodies and other structure proteins are all made. Why all these important functions are provided by almost exclusively by proteins? The answer is proteins can become structurally diverse. DNA. Try to imagine the DNA structure. It's always boring, double twisted shape. RNA always like more skinny, single twisted structure, more or less. In the case of RNA, yeah, I have more details, but I will shut up right now. But basically, these nucleic acids are highly predictable, okay, repeated structure. Carbohydrates too, although in the occasion of oligosaccharide, you can generate some like little cutie diverse shapes so that with which you can recognize its different surfaces, but that's all. Carbohydrates are also basically kind of highly predictable repeated unit of structures mostly. Lipids, same thing. Same repeating unit of structure. No excitement. But in the case of proteins, yeah, you can enjoy so many different exciting structures. And nowadays, actually, these protein structures are entering protein structure determination, the area of research, are entering whole new horizon with the help of, once again, guess who? Artificial intelligence help will actually significantly elevate this structural determination. In fact, actually, several new versions of this coronavirus vaccines were actually generated by, with the help of this AI protein structure, the prediction tool. Without it, it wouldn't have been possible. Okay. So, anyway, the point is, this protein structure is so important, like this. Many different structure can come because of this diversity of amino acid. 20 different amino acid you can have arrange, assemble, pick up combination. So you have so many different scenarios. And that's it. So as consequences, you can have so many different diverse functions with the proteins. Now, this is a structure of any particular one given enzyme's internal structure where that such magic, the enzyme catalysis occur. We call it an active site.
Enzymes are proteins
Or
that
should.
carry out
Oh, please shut
chemical
up.
They have two special features. First, they are highly specific.
How can I?
Each enzyme acts only on select molecules called its substrate. Second, enzymes are catalysts.
Okay, I got you. So can you shut up now?
And they are unchained reactions.
Shh. Okay, good. Sorry. So this enzyme are biological catalyst, as we have already went over. What do you mean by actually biological catalyst? So as a catalyst, it is not something that got hurt or spent during the reaction, but it's there as a bystander. It stays there. Yeah. It's not being consumed, but simply it can speed up the reaction. That's it. And why do we need, if we ask, why do we need the help of this enzyme for any given chemical reaction? If you ask, speeding up is something, yeah, of course. So we can finish this one particular, the chemical reaction within a reasonable period of time. That's very important. And especially enzymes, you may think, yeah, of course, enzyme would be specifically required if we consider that there are two different kinds of chemical reaction. One is spontaneous and the other one is non-spontaneous. Remember the nature of a chemical reaction. One is spontaneous. The other one is non-spontaneous. How do we distinguish these two chemical reactions? What's spontaneous reaction? Kind of a natural thing. It will occur regardless. Any spontaneous reaction will happen eventually. Okay? So no matter what, so you don't worry about. It will happen. Okay? Sometime. Don't get nervous. It will be there. It's a spontaneous. Why? Why is any reaction spontaneous? Let me ask you. Why would any reaction be spontaneous? So simply put, maybe I can draw this line to describe this spontaneous reactions characteristics like this. This would be spontaneous. Like let's imagine that you are on the hill, on top of this hill. Okay. And if you try to, let's say there is a rock on top of the hill, and then you try to roll this rock to the bottom, it will be so easy, isn't it? You just all you gotta do is simply roll this thing, and then it will go downhill so easily. That's kind of a nature that we can describe the characteristic of spontaneous reaction. Is there any other way to describe or characterize this any spontaneous reaction? Instead of this rock thing on top to the bottom, if we try to define this y-axis area as what? The energy. This area is like amount of energy. And this x-axis as a like a reaction progress, then probably that's another way of describing this any given spontaneous reaction. What do you mean by that? Any spontaneous reaction, would any spontaneous reaction release the energy or require the energy at the end of the chemical reaction? If this was the progression, the direction of the reaction, and if this y-axis was the energy level, so this would be the start of a chemical reaction, and this would be the end of the chemical reaction. What do you say? What do you say? And if this is a kind of a diagram describing any general spontaneous reaction, what do you say in terms of energetics? Spontaneous reaction would spontaneous reaction release the energy or absorb the energy? Release, right? Everybody agrees. Because in the beginning, this reactant, the material entering the chemical reaction, has so much energy like this. But at the end of a chemical reaction, their energy level has gone down to this level, which means where has all this energy gone? They were all released. And so any reactant now as a consequence of this chemical reaction became what? More stable. Stability is always something to do with the energy level. If you have high energy, remember the radioactive isotope. Why the radioactive isotopes was so unstable so that they release the energy? Because they have so much high energy. So to become stable, to become more stable is always the chemical nature of everything in the universe. So if you have this so much energy, then they will do anything, everything to become more stable by after releasing this energy. That's why this type of reaction will be spontaneous. You don't have to do anything. They will do this. They will go toward this area. Of course, we can describe this thing as more complex ways, introducing some concept of, so to speak, entropy or anything, but not here. So simply energy-wise, we can describe such the characteristics of a spontaneous reaction. On the other hand, then non-spontaneous reaction would be what? Totally opposite. The chemical reaction would be something like going toward this uphill in terms of energetics. Starting material's energy level will be much less than the final product energy level. How can that be possible? That's why we call such reaction as a non-spontaneous. So any non-spontaneous reaction will be like this, going like this, if this was the direction of the reaction. So how can you make any non-spontaneous reaction actually happen? Provide the energy, because it ended up having more energy. So without providing, in the beginning, without providing the energy, it's not going to happen. That's the nature of non-spontaneous reaction. And between these two different kinds of chemical reaction, which one, spontaneous or non-spontaneous, which one would require the intervention of enzyme more? Probably non-spontaneous reaction, because we can intuitively think that this type of reaction would be difficult, so probably we need some chemical catalyst. Yeah, that's kind of our like common sense would explain. But in reality, in our cell, in body and the metabolism, both spontaneous and non-spontaneous reaction definitely require the enzyme catalysis. Why? And how? And if you think a little more carefully, what do you mean by that in the case of a non-spontaneous reaction, what's the actual role of enzyme? Do you mean by that enzyme, if there is an enzyme, then this whole pattern, the profile, reaction profile of non-spontaneous can be changed into something like this? Well, what do you mean by that? And in the first place, why would such a natural spontaneous reaction ever even require the enzymes? What's the reason? Is what we are going to have a look at the as the last part of today's lecture. So once again, this enzyme is there. It's not consumed but simply speed up the chemical reaction. So why does a spontaneous reaction need an enzyme? Is this spontaneous reaction, another name for this spontaneous reaction is catabolic reaction, because these catabolic reactions are always release the energy, and catabolic reactions are always moving from more complex to the simpler structure, like breaking down reaction. So, as naturally, it will generate the energy. So it should occur by itself, but because of this something weird thing like activation energy, even the spontaneous reaction cannot occur, okay, by itself without the enzyme. Funny. So this diagram, this is a progressive reaction and this is the energy. And obviously the start and end energy level is different in that overall it should be spontaneous or non-spontaneous. This type of reaction. Spontaneous, right. But something weird. What is this? Like this camel's hump? What is this? This is what we call activation energy. There is some kind of activation energy. I can explain what it is in terms of the enzyme structure, like substrate enzyme structure, but forget about it. Okay. Simply, we can imagine kind of the activation energy is just sort of like some kind of a hump on top of the hill. That will be just enough to understand the nature of this activation energy. In other words, if you had a rock over here, in reality, whenever there is this kind of a hump represented by activation energy, it will not be easy to roll this rock to the bottom because of this presence of this hump. You have this kind of hump. You have to overcome this hump to roll this rock down. Okay? So to do so, you need to put some energy, right? It will be hard. But enzyme can lower significantly this hump. That's what the enzymes try. Then if in the presence of the enzyme, this whole insurmountable high hump can be reduced, like manageable, to the manageable height. So now it's easy. Okay, I can do that. So that's basically the job of enzyme needed for this any spontaneous reaction. This is really, really childish, elementary version of the explanation for enzymes requirement for spontaneous reaction, but still it works. Yeah. So it can reduce the activation energy. The bottom line is conclusion. Why any enzyme is required for spontaneous reaction? Because enzyme can reduce the activation energy. So it practically it is like enzyme can make this any spontaneous reaction practically possible. One case in point, given the example, maybe I already have mentioned this because that's another one of my favorite the examples, like breaking down glucose. Whenever you eat some food, inside of your cell, the starting point is breaking this glucose down into. So let's say glucose is a 6 carbon. 1, 2, 3, 4, 5, 6. The first step is what you gotta do is okay, 6. The first step is to reduce this into 2 halves, like 3 carbon structure. It's very simple. This whole thing. That's the first line of reaction that is going on for any food material like containing glucose to we digest and then extract the energy. This simple reaction is actually catalyzed by 10 different enzymes standing in line and handing over to next guy and then make eventually this 2 part. Okay? Like a big baguette, you are breaking this whole baguette into 2 halves. That simple reaction requires 10 different enzymes. Wow. Overall, is this spontaneous or non-spontaneous reaction? Spontaneous, because it's catabolism, breaking down. It always should be a spontaneous reaction. However, without, okay, so you thought Okay, this is a spontaneous reaction. Why would I need any enzyme? You go out. You are all fired. So you just kick all this enzyme out and then let this, yeah, this should occur naturally. So let's be a little bit more patient. After eating our meal, they will be naturally broken down into these two. How long would it take? 5,000 years. Without the involvement of this enzyme, this breakdown of this one glucose molecule into two halves will take 5,000 years, during which everybody probably would starve to death. With the enzyme, it can be done in the matter of several seconds, right? So that's the whole big actual real differences and real occasion why enzymes can make differences in chemical reactions. And simply put, how? When we ask how, by doing this. Okay. The reason why it takes such like 5,000 years is this is too high, so they will keep trying. So it will take so long time and eventually make this happens. Kind of an analogy. Not exactly true, but try to explain in a very simple way. Now, so that's we can put this thing aside. So now everybody is okay, happy with the reason, the explanation for this spontaneous reaction and the enzyme requirement, right? What about this non-spontaneous reaction? Yeah, like as I mentioned earlier today, non-spontaneous reaction, we can intuitively kind of anticipate that, yeah, that's something unnatural, so we need some kind of special magician to make this thing happen. So that's, yeah, oh, no wonder we need an enzyme. But if you were asked once again on how, then probably will be difficult for you to explain clearly. So this backward reaction, from starting from simpler individual molecule, trying to build the more complex reaction, we generalize such reaction as an anabolic reaction. And obviously, those kind of reaction would require the energy input. And on that occasion, we call this that type of special activity as the term. There is a specific term describing the role of enzyme in such anabolic reaction as energy coupling. So the enzyme's job is like acting as a matchmaker. You find one, okay, your task is, your objective is to make this non-spontaneous reaction happen. How? Non-spontaneous reaction requirement is you have to provide the energy in the beginning. So how can you provide the energy? It's simple in conceptually speaking, but how? Mechanically speaking, how can you provide the enzyme, right? The enzyme can bring two different reactions together and make it happen at the same time in their home ground. We call it an active site. One, this non-spontaneous reaction, which is our objective, and to drive this non-spontaneous reaction, you also invite at the same time another downhill reaction. Like very famous such downhill reaction would be ATP hydrolysis. That's why ATP is used as a general, like a battery-like energy form. ATP, you break down ATP into ADP, then energy will be released, right? So you couple these two. Your objective, your uphill non-spontaneous reaction, your objective. How can you make this thing happen? Bring this ATP together. Hey, why don't you come over here? And then let these two guys meet together. So by breaking this ATP, energy will be released, and those surplus energy will be funneled into this energy requiring uphill non-spontaneous reaction. So overall, if you sum these two different reactions, overall reaction will become downhill because you have a huge surplus over here and you have this deficit, energy deficit over here. If you add up these two together, this surplus will be greater than this deficit. So altogether, you have some surplus in the energy, making this overall reaction downhill, spontaneous. That's what the enzymes are. That's why most of these enzymes, that energy requiring enzymes, actually use ATP as the energy source. By breaking this ATP down, so that energy will go into this. Yeah, sounds plausible. Nice explanation. Right, it is. However, this explanation itself is not actually really correct. It's kind of very simplistic, elementary version of explanation, but once again, it works. Okay. So if we accept this as a what? Energy coupling, that's what we can understand at the level of general biology, the enzymes role. So what's the bottom line? Enzymes are needed for both spontaneous and non-spontaneous reaction for different mechanisms. In the case of a spontaneous reaction, they reduce what? What was it? Activation energy. Okay. In the occasion of a non-spontaneous reaction, it's not something that the activation energy matters anymore. Okay. Activation energy will be still there, but more importantly, what you have to overcome is this deficit, energy deficit. You have to provide the energy. How? How can you fund this energy requirement? By luring another this poor guy like ATP. Spontaneous reaction. So making these two reactions happen together at the same time, then overall reaction will be spontaneous because of this coupling. That's what this energy coupling definition. Okay? Okay. Right. I have no energy for jumping into new chapter. Probably you are happier than me trying to go into unnecessary new chapter before taking final exam. So I know this the what we have covered so far is very minimal. But anyway, still I will try to come up with some questions or over the up until now what we have covered, and then once again, like I said, I will just put up them in the snowboard by next Thursday, right? So okay, so good luck. And next Friday, still there will be a lecture over here, kind of a makeup lecture, but attendance is not mandatory. So for next week's lecture, I'm going to go over my makeup lecture, but there won't be any scrap paper requirement for turning in because it is a makeup lecture. If you can come, you are more than welcome to be here. If you cannot come, so be it. Okay, so bye bye.